Welcome back, everyone. So you have finished your first 4 chapters. You've taken your first exam, and now we are jumping into the second unit of class. We are going to be talking about how cells do the things that they're doing. How do they get energy? How do they multiply? How do they copy their DNA? How do we kill them? How do we use antimicrobial drugs? That's kind of what we're looking at this next unit we're talking about. So in chapter 5, this tends to be everyone's least favorite chapter. So I hope you take that as a warning that it's a little intense, but just give yourself some rigor to really buckle down and work on it. And also give yourself some grace if some of this is just a hot mess to you. It takes a little bit of time. It takes a lot of time to be able to understand some of these concepts. My very first semester of college, really, it was my first time to really have a good, solid biology class. My high school biology teacher was a football coach, and so I learned to say, yay, go team, more than I learned to do biology. So I got to college. I really didn't know what was going on cell-wise, but I knew I wanted to learn about bacteria. So I took my first class, and we start talking about metabolism, and I'm doing okay up until we get to the electron transport chain. That entire semester, I had no clue what was going on in there. Then I took my next biology class next semester, and we get to the electron transport chain, and, bits and pieces of it I could put together. Still, the whole process was kind of a muddy water. I could kind of see pieces of it, but definitely couldn't see to the bottom of the pond. I was probably a junior in college before I really started being like, oh, I get it. So my goal for you is not for you to take this chapter and leave this chapter thinking this all makes perfect sense. Cells are completely obvious how they function. My goal for you is just to be able to at least understand a few details. Today's topics that we're covering are probably going to be a little bit more on what you're able to understand. It's once we start talking about glycolysis and the Krebs cycle and the electron transport chain where it might start falling apart. And so there is a file provided for you to kind of chart through where each step is happening, what kind of energy is being made, and what else is kind of going on. If you can just follow through that chart, you have enough understanding to be okay for this chapter. Okay, so let's jump in. Let's start. So metabolism. Metabolism is just everything that's going on in that cell. All the chemical reactions. Everything that cell is building up. So it's time to build a new piece of DNA. It's time to build a new protein. It's time to build a new phospholipid. Everything it's building. Everything it's breaking down. It's taking in some sugar, breaking down that sugar, making some nice energy, brought in a little piece of fat or a little protein, broke it down, got some energy. Everything it's building up, everything it's breaking down. Metabolism can be broken into 2 subcategories, catabolism and anabolism. So I think of a cat and I think of a little girl named Anna when I'm thinking of these. So imagine that there is a block tower being built. If you had to guess, it is probably not the cat building up that tower. It is probably the little girl named Anna who is building her tall block tower. If you imagine that cat is probably going to come along and start just pawing at it and just break that tower down until it falls into pieces. So cat, like catabolism, is breakdown. It is taking something big and breaking it down into smaller pieces, and this will produce energy. So taking in a molecule of sugar, breaking it down to carbon dioxide, and giving energy. These are associated with hydrolytic reactions, because that is what breakdown is hydrolytic reactions. Anna, who is building the block tower, just like anabolism, is using energy and those little pieces to build up a large molecule. So it is taking little individual nucleotides to build a strand of DNA. This is associated with dehydration synthesis. This requires energy. That energy for anabolism is going to come from catabolism. So catabolism and anabolism are going to be linked to each other. And generally in our cells, whether it's a eukaryotic cell or a prokaryotic cell, our cells are not just going to take something and in one big step, just poof, you've gone from little pieces to a big molecule, or you've gone from one big, huge thing to poof, individual little pieces. It's going to be more controlled and very carefully and slowly, gradually building up from one piece to a larger piece, or from a larger piece down to the individual pieces. Those sequence of steps are called a metabolic pathway. What kind of metabolic pathway a cell is able to do is going to be determined by what enzymes are there. So it'll be able to do one reaction because it has the enzymes, or won't do the reaction because it doesn't have the enzymes. And those enzymes are going to be encoded by genes. So different species or different cells that have different genes in them will have different enzymes and be able to do different things. So sometimes we can look at a cell, if we don't know what kind of cell we're looking at, we can look at what kind of reactions it's able to carry out, what kind of metabolic pathway it's able to do. Is it able to break down lactose? If it's not able to, it's lactose intolerant, or is able to use lactose. We can then see what kind of enzymes it has, know what kind of genes it has, and that can help us identify what kind of bacteria we're looking at. Okay, so I mentioned catabolism and anabolism are linked together through energy that they're using. So if we start with some simple molecule, so something like glucose, some amino acids, some glycerol, some fatty acids, just these individual little building pieces. Anabolism is going to put these together to build complex molecules. So glucose put together to make starch, amino acids put together to make proteins, glycerol and fatty acids put together to make lipids. This is anabolic, building up larger molecules. This is also going to require energy. So ATP is the energy source. ATP is going to be broken down to ADP and little pieces of phosphate. And now you have big old stuff. Then that cell can come and eat these big old things and do catabolism. And catabolism is going to take these big molecules and break them down to smaller ones. And as it breaks those down into smaller ones, it'll create ATP. So it'll take that ADP and use the energy from breaking down the molecules, transfer that energy from the molecules into ATP. So you just have this cycle of eating something, getting energy from it, using that energy and those building blocks to build up what you need, using ATP as that intermediate. Okay, so metabolism is important for a lot of reasons. It allows bacteria to grow and cause disease. Probably very important for most of you because many of you are going into some sort of medically related field. Microbes growing can cause food spoilage. So, if you've ever had your milk get a little bit sour, or, your sandwich in the back of the fridge has gotten a little bit of mold on it, that's thanks to the metabolism of the bacteria growing there. Probably also important, I'm going to guess that most of you eat food. So both of those are pretty important to us. But metabolism also can be really important because many of these pathways can be beneficial for us. We can harvest microbial metabolism to produce antibiotics. We'll talk about that a little bit more in chapter 20. But it's really interesting that a lot of the antibiotics we get are from other microbes that are growing. We can use metabolism to produce vaccines, to produce vitamins, to produce enzymes that allow us to stay healthy. Okay, so one thing, I'm going to play this, muted though. So the collision theory is saying that molecules all around us are moving all around. So just like in this little picture, these molecules are just always moving. They're constantly moving. They're constantly running into each other. And sometimes when they run into each other, they hit with enough force that they can make new molecules. They can make new bonds. They can break old bonds and make new bonds. The amount of energy it takes to break the starting molecules and rearrange them into the ending molecules is called activation energy. And the reaction rate is going to be how frequently those collisions occur. So they have to be occurring with enough energy and in the right orientation to have the reaction happen. Reaction rate can be increased by using enzymes. Enzymes will put those molecules together in the orientation that makes them really line up and work the best. Temperature. So temperature will actually make those molecules move a little bit faster, so they run into each other a little bit harder. So that means they'll have a little bit more energy when they run into each other. Pressure can increase reaction rate. So pressure is when you just push those molecules closer together. So as they're closer together, they're going to be running into each other more frequently. So the reaction rate will increase by that. Catalysts are things that will speed up a chemical reaction, but they are not altered. Okay, so that's important. They speed up the reaction. They make that reaction happen faster, but they are not altered. So at the start of the reaction and at the end of the reaction, your catalyst is unchanged. It is identical. Enzymes are types of biological catalysts. Enzymes are going to be proteins. And so when I ask you what is an enzyme, you can tell me they are proteins. They speed up chemical reactions. They are not changed themselves. Enzymes are also specific. So enzymes will act on a specific substrate to lower the activation energy for that specific reaction, but they won't affect other reactions. Okay, so like if you have the enzyme to break down lactose, you can break down that molecule of lactose into 2 individual monosaccharides. If you don't have the enzyme to break down lactose, another enzyme can't come in and do that job. You just won't be able to do that reaction. When a substrate and an enzyme act together, the substrate will fit into the enzyme's active site. The active site just means that little pocket on an enzyme where the substrate can fit. And that forms an enzyme-substrate complex. That is a term that you need to be comfortable with. When the substrate is interacting with the enzyme in that enzyme-substrate complex, that enzyme will kind of help push it, twist it around, give it a little bit of pressure to transform that substrate, break the old bonds, form the new bonds, and create products. The product is the finished product. It's what you end with. And those products then will be released from the enzyme. And the enzyme is unchanged. Okay, no change to your enzyme. It can go back and react with other substrates. And some enzymes can work, maybe 1 reaction a second. Some can react hundreds or even thousands of reactions a second. So here is the reaction happening without the enzyme. And look at all this energy. This is your reactant. A and B are bound together, and then they're breaking apart. So we're breaking apart. We know that this is a catabolic reaction. Remember that term. But it's going to take all of this energy to make it happen before it can break apart. Even though you are releasing energy overall, you have to start with energy to make it happen. And that's a lot of energy that if you don't have the enzyme, you are not breaking that molecule apart. But when you put in the enzyme, then you only need this much. Only that much energy, and then it'll break apart. So this enzyme is just going to kind of help twist that AB molecule apart until they release as those products. And the enzyme goes right back to find another AB molecule and do the reaction again. Within an enzyme, there are different pieces of it. So there is an apoenzyme and the cofactor. An apoenzyme is the protein portion of the enzyme, but it's inactive on its own. So even though it has the protein there, it's not able to really figure out how to do anything by itself. It needs a cofactor or a coenzyme to come in and activate it. Cofactors and coenzymes are technically different, but I'm not worried if you can tell those words apart. Cofactor or coenzyme, either one of those, if you know, that is a non-protein piece that comes in and activates the apoenzyme. So the apoenzyme by itself doesn't do anything. But when that cofactor or the coenzyme comes in, it'll activate the apoenzyme. Some coenzymes we'll talk about, NAD+, NADP+, FAD, and coenzyme A. Those are some things you're going to hear talked about in this chapter. Those are all coenzymes. They're going to activate the apoenzyme. When you have the apoenzyme and the cofactor or the coenzyme together, you have a holoenzyme. So I think of whole, like WHOLE, whole, like complete. And holoenzyme, it's the whole thing. You have the apoenzyme and the cofactor together. And as a holoenzyme, it is going to be active. It'll be going out and carrying on those reactions with a turnover number that could be between 1 and 10,000 molecules a second. So doing all those reactions again and again. So here is your apoenzyme. Sweet little protein piece. Can't do a thing by itself. Here is your little coenzyme. Coenzyme and the apoenzyme can fit together. And here you have the holoenzyme, or the whole thing. As a holoenzyme, it is now active. So the substrate can come in, fit in right here. That means this is its active site. That's where it can fit in and carry out the reaction. Okay, so let's start looking at here. Here's your holoenzyme. The substrate is able to fit here. This is the active site. This is the enzyme-substrate complex. You have the enzyme and the substrate together. They give it this special little hug that just breaks that little enzyme, or the enzyme breaks that little substrate apart. And now you have the products. The products are different from the substrate. The products is what you end with. And those products are released. But look at this enzyme. It is exactly the same as what you started with. The enzyme is a protein that makes a reaction happen, but itself, it is unchanged. So this little protein now goes right back to the beginning, finds another substrate, and carries out the reaction. Goes back, carries out the reaction, until all of that molecule is broken down. So at this point, you can define catabolism and anabolism. You can describe how ATP is an intermediate between catabolism and anabolism. You can describe to me what collisions have to do with reactions, what reaction rate is. You can tell me, how does an enzyme affect activation energy? You can tell me, how does it affect a reaction? You can tell me what is the holoenzyme, what is the cofactor or the coenzyme. All of those terms we just described. Okay, come back for part 2, and we're going to start talking about some energy going on in our cells. See you later.